Let me give you one more example. And this is work that will relate to something Jeannie Lee was talking about with long non-coding RNAs. And in particular, it's been collaborative work with John Rinn and Mitch Gutman and Aviv Regev, all connected with this project. And in one particular part I'll particularly note, with P53, collaboration with Tyler Jacks. So the epigenome. Jeannie was talking about the epigenome and was saying that she might be the token person talking about the epigenome. Well, I'll be another token person talking about the epigenome because I think it's an important thing. And you have all these marks on the epigenome. Sequencing has made it possible to rapidly localize those marks by using an antibody against a particular histone modification or other things, pull down all the DNA that's associated with a histone with modification, sequence it, and look for patterns of where all different kinds of marks are. Well, one of the patterns that emerges is that an actively transcribed gene has histone 3 lysine 4 trimethylation around its promoter, and histone 3 lysine 36 trimethylation along the length of the elongated transcript, the transcribed region. This is a signature of a gene. So you could start looking for genes this way. Now, why do I care? I care because there's been this controversy in the literature of genomics for quite some time about how much of the human genome is really truly transcribed. On the one hand, there are these papers that are saying every nucleotide of the human genome is transcribed. But to do that, you have to get down to very, very low levels, and it's very messy, and I don't want to think the genome works that way. On the other hand, you have beautiful work like Jeannie Lee describes with EXIST and T6, beautiful, kosher, long, non-coding RNAs that show nice properties. It's work of hot air, but there's only been a dozen to 20 really well-characterized examples of long non-coding RNAs until the past couple of years. Are there really a lot of these things? Well, chromatin signatures can tell you in an unbiased fashion. Take a tissue, make a chromatin signature, start looking at, looks like a gene, looks like a gene, looks like a gene. Is it known protein coding gene? Cross out the known protein coding genes, find new ones. When you do that across a bunch of tissues, you find 5,000 instances of long intergenic non-coding RNAs, a whole collection of these things, about a quarter as many as there are protein coding genes. They're a small portion of the genome, mind you. They occupy less than 1% of the genome in terms of the mature transcripts that are made, but they're highly conserved. Well, not as highly conserved as protein coding genes, but they're well conserved. And they're as conserved as exists in hot airs and things like that. Their promoters are very well conserved, et cetera. What do? Well, take the gene expression patterns. Get the gene expression patterns of all these non-coding RNAs, in this case with about 400 of them. Compare them to the gene expression patterns of protein coding genes and start keeping score. Here's a non-coding RNA and its gene expression pattern across a bunch of tissues. Let's see how it compares to genes in the NF-kappa B pathway. Ooh, well correlated. We'll give it a good score. How about fatty acid metabolism? No correlation, give it a mediocre score. P53, anti-correlated. We'll give it blue. And you can do that for 1,600 different pathways. And every non-coding RNA then gets some identity as to how it's being expressed. You can then cluster the link RNAs against these functional gene sets and begin to say, if I've got proteins who have known functions, I can begin to assign guilt by association functions to these non-coding RNAs, and I can find things involved in that allegedly could be involved in immune response, brain processes, muscle development, gametogenesis, or P53 related. So let's take P53 related, since this is a cancer meeting. So you can then check that these things that show across tissues correlations with the expression of P53, in fact, show appropriate induction when P53 gets induced in various different settings. And here, of course, is all collaboration with Tyler Jacks's lab, where you have P53 wild type and knockout cells from appropriate mice. And you can see that they're really induced. The genes that show this induction have in their promoters, these non-coding RNAs have in their promoters, P53 motifs. They look very kosher. And here's one in particular. It lives nearby the P21 gene, and we'll give it the name lincRNA P21 because it's nearby. What does it do? Well, P53 is known to activate lots of genes, and now link P21 gets to be one of the genes that P53 activates. It also is known to repress a lot of genes. How it represses genes is poorly understood. If you knock out P53 and you knock out link P21 separately with shRNAs, you can ask what effects. Knocking out link P21 affects the apoptosis phenotype, but not very much the cell cycle phenotype in these cells. But when you look at gene expression, you get a very interesting clue. Link P21, when you knock out this non-coding RNA, affects a set of genes that is, largely speaking, a subset of the genes affected by P53, and particularly the ones that are repressed in the P53 pathway, leading to the hypothesis that link P21 is functionally acting as a repressor in this pathway. So is it involved as a repressor, and if so, how? Well, if you take link P21 and biotinylate it and take a control link RNA and you incubate it with cell extract and you pull down what proteins come down with it and run them on a mass spec, you find HNRNPK, the heavy nuclear protein, or ribosomal nuclear protein K. You can show, and John Rinn in his lab particularly have shown that it binds link P21 in vivo and in vitro. It binds the most conserved regions of this non-coding RNA, and it binds directly to P53 repressed genes. You can show that the link RNA is required for the binding of the protein to these promoters, and that the binding is necessary for the P53-mediated apoptosis, at least the full measure of P53-mediated apoptosis, which presents a hypothesis that link P21 collaborates with HNRNPK to repress genes in the P53 pathway. Role in cancer, could this be a mutation? Could this be a target of mutation in cancer? Nobody's ever looked. In our exome screens, we've never had it on there before. Now we're beginning to add these link RNAs because this could potentially play a tumor suppressor role. What about more than just this link P21 gene? Well, it turns out that about a quarter of these link RNAs bind chromatin-associated proteins. So a quarter of them bind polycomb, in fact, and about another sixth or seventh or something like that bind other chromatin-associated proteins. And so it's not unreasonable to think that this is a general theme about these link RNAs, is that a significant fraction, of course I should say, exist is a classic example, and hot air is a classic example of non-coding RNAs binding, but now this suggests that perhaps a quarter of that whole universe are involved in that way. And you can functionally test that across the board by asking, if you knock out polycomb and you knock out the link RNA, which genes are dysregulated? We did that for the P53 link P21 case. You can do it for a bunch more cases of link RNAs associated with chromatin proteins. You can look at the gene expressions. You can say, are they similar, or are they what? And I won't go into the details. And the short answer is the majority of link RNAs that are associated with chromatin binding proteins, when you knock them out, have gene expression patterns that are related to, in a highly statistically significant manner, the gene expressions of knocking out those chromatin binding proteins. So the hypothesis is that there are a large number of non-coding RNAs that function as repressors, perhaps in different transcription factor pathways. They collaborate with different proteins to localize in ways still mysterious to us how this gets done, that they get to the right place, but act as repressors. And there may be a whole layer of interesting regulatory biology that sits there, and at least with the P53 example, potentially has implications for cancer. I'm going to, in the interest of time, skip over the last bit, which was going to relate to Jeannie's 3C, but no time for that, and simply say, well, maps can teach you lots of things. There's lots of things still to learn about cancer, and I think the collaboration between cancer and genomics is about to just skyrocket because the costs of producing data are dropping so rapidly that I will predict that within a few years it will be a routine thing for anybody characterizing any cancer model in a research laboratory to at least get the whole thing sequenced across its exome and pretty soon after that across its whole genome. And I would hope that at some point within the decade to come, it'll also become a pretty routine thing in clinical laboratory settings as well. Not that it will immediately answer everything, but it will provide us with a plateful of hypotheses and a tremendous amount of data, some of which will be clinically useful. I just want to stop by acknowledging the wonderful collaborators on the broad topics of genome sequencing at the Broad and cancer genomics generally, but particularly the multiple myeloma project, on which Todd is the senior author of this paper, and Mike Chapman and Mike Lawrence, and the Link RNA project that's been an ongoing collaboration amongst John Rinn, Mitch Gutman, Aviv Regev, and then particularly with this new paper that is in press now with Tyler Jacks, and I won't have talked about that last topic there, and I will stop.